Hi, everybody. I'm going to continue with what we weren't able to do in the lecture, which is to discuss character-based methods, at least briefly. And while I have identified some aspects of the slides that are really not perfect, I'm going to continue to use the slides here just in the interest of time, and I may go back and make some additional videos to try to clarify some of the things that may be a little bit difficult to cover properly using the slides I have here. So the three major approaches to character-based methods are that we're going to discuss, or the approaches described here. So the first method is parsimony. And parsimony, we've actually seen before. Parsimony is what was used by Margaret Dayhoff when she did alignments in order to eventually identify these accepted point mutations. So she did the alignments first using parsimony. From those parsimonious alignments, she then picked those substitutions which were accepted in evolution rather than all of the substitutions. So the idea of parsimony is that we're seeking a tree that has the minimum number of changes that are still compatible with the data. The idea of parsimony is that the best model of evolution is the model that requires the least of it. And in some sense, you can think of this as kind of similar in a way to Occam's razor. Parsimony has a property where a parsimony inference will often infer several or perhaps even many equivalent trees that are equally parsimonious and therefore cannot be distinguished. Now, the second model is that we're going to discuss is a statistical model, and that's a model called maximum likelihood. Maximum likelihood is used far outside of just phylogenetic tree inference. Maximum likelihood is used all over statistics. It's a very powerful and widely used model. And the idea is that we frame a problem in terms of a probability, or it's very much related to a probability called a likelihood. And that likelihood is a conditional likelihood. That is, what is the likelihood of the data given a model with some parameters. And then what we can do is modify those parameters and do that until the likelihood can't get any better. So that's what that is. It's a little weird because we're trying to, we frame it in such a way that we are looking at the probability of observing the data we have in hand. Bayesian analysis kind of turns it around, and it's a probabilistic method and says, well, given the data, which tree or trees has the greatest likelihood, or the highest probability is usually how it's termed in Bayesian terms. Maximum likelihood and Bayesian analysis are related, but very much not the same thing. They come at the approach with very different kind of philosophical approaches that have real world consequences. So let's go through these in a little bit more detail. You've seen parsimony before. It comes up in PAM matrix construction, and we're going to try to find the tree that requires the least amount of evolutionary change. That's considered the most parsimonious. Now, there are trees that are found through a variety of computer science algorithms. Classes of algorithms are described here. And if you've taken a class in algorithms, you may have run across at least the second method here, the idea of branch and bound. This class, we do not cover these algorithms in detail. So while it's nice to know that these algorithms are part of parsimony work, it is not necessary for you to know how these things work. One of the characteristics of parsimony, which its critics really don't like, is that parsimony has a particular susceptibility to a problem called long branch attraction. So the idea is very often found when a particular character evolves very rapidly. So maybe one character or a subset of characters evolves very rapidly along a particular branch in evolution. Now in DNA, there are only 4 letters, right? So we can see only one change, and there aren't that many options. There are only 4 options. And so a rapidly evolving site might be inferred to be unchanged. This should be a D. when in reality, it may have changed twice or 3 times or 10 times. And so we will remember the nucleotide substitution models where we convert the number of changes that we see into some evolutionary distance. Well, parsimony, in this case, for the long branch, in the case where a character is evolving rapidly, that character, we may underestimate the evolutionary distance because we're unable to see it. And this can lead to statistically inconsistent results. Parsimony, maximum parsimony, is particularly prone to this behavior. And now when we look at problems in statistics, if you've ever talked with somebody who does statistics or even joked about it, there's a common joke, they'll say, you need more data. And that's true for a lot of problems. Getting more data helps a problem become easier to solve or less likely to go wrong. But that's not the case for long branch attraction. The more data, the more likely it is to be wrong. So it's really not a good thing to be subjected to. There are various methods to detect and try to resolve issues when long branch attraction does occur. All of these methods are imperfect, but you should be aware of it. Now, maximum likelihood has this very statistical approach. It has some data. So the data is, we have a multiple sequence alignment, we have the characters in that multiple sequence alignment. And then we have a model. And remember, all of these trees are models. So a model may have parameters, just like a linear regression model has parameters, a slope and an intercept. Well, tree models also have parameters. And our hypothesis is that the model, this evolutionary model, is a tree, and it's going to be in a tree-based shape. And so what we can do is, if we modify the parameters, we will get different trees. And we modify those parameters until, and for each one we calculate, what's the likelihood of observing our data given this particular tree? And we compare, and if one tree gives greater likelihood than another, then we keep the one that gives greater likelihood. And we try to maximize that. So we basically keep modifying the parameters until we can't anymore. And at that point, that's the maximum likelihood, and then we're done. So if parsimony gives many equivalent trees, in maximum likelihood, we might see something like this. Okay, so I'm going to draw here an L. It's a very poor L. Whoa, that's an L, sort of an L. Let me try drawing that again. I'm using a different annotation system for this. Okay, so it looks like this. That's a likelihood L. Okay, so if we have if we imagine a graph, and then we have a bunch of parameters, and the parameters are typically called they're called theta. That's all the parameters. And so we keep modifying theta, we get different trees, and we're going to get a likelihood. And we might find that the trees that we get have this kind of, now it's going to be much more complicated because theta's many different parameters. So it won't just be one. And then we'll see, maybe we might draw kind of a curve here for that. And then this becomes the maximum. Somewhere here becomes the maximum. Now, when we have that maximum, then that becomes the tree that we choose. But if you'll notice here, even though they're not equivalent, like in the case of parsimony, there may be many trees, many values of the parameters that give trees that have very similar likelihood to the maximum likelihood. And that is very much the case. It's often the case. Where'd the mouse go? There we are. Okay. So, yeah. Now, for a lot of methods, actually, it says here for parsimony and maximum likelihood, but this is true for many other methods, Bayesian methods, and even distance-based methods. We use resampling to estimate the reliability of a tree. So what do I mean by the reliability of a tree? Well, if we have a tree that is, let's say here, okay, we have a tree here that is you know, we have a tree that looks like this, and we have a tree here. And so that's a tree. Now the question is it that tree, or is it one of the other trees that is possible with these three things? And so the reliability is, how often do we get the same tree? So if we have, is there something about our data that's particularly pushing us to this tree? And if we had slightly different data, we might have a different tree. So the methods that are used for this are resampling methods. The two most common methods are the bootstrap and the jackknife. And the bootstrap is the key one that we're going to discuss. So and what we do is we basically, we have our data, and we create an artificial data set that's the exact same size as our original data. And we do it by replacing or drawing at random a character out of our original data set. That's what I mean by saying that a bootstrap is a little bit like a lucky draw. What we do is we take all of the characters and we put them, you know, write them down on a slip. Okay, character 1, character 2, character 100. And then we reach into, we put those slips of all of the characters into a bowl. We reach in, we pick out a character and we say, okay, this is character 38. And character 38 becomes the first character of our simulated data set. And then the key thing is the slip that we just took out is replaced, put back in the bowl, so we can draw it again. In fact, we can draw it the next time. The second character could also be character 38. If we didn't take it out, if we did sampling without replacement and we sampled all the characters, we would get all the characters. They would just be in a different order. But the order in this case doesn't really matter. It's which characters there are. So in a bootstrap sample, we are doing this thing called resampling with replacement. Okay, that's what it's called, resampling with replacement. And the with replacement part means that we put the slip back in that bowl. Okay, so we can draw the same character again, and some characters may never be drawn. So that when we do that one time, that becomes one bootstrap sample. And then we take that bootstrap sample and we re-estimate the tree, exactly as we did with our original sample. Now we have a new tree, and we can compare those trees. Are they the same? Do they differ at one position? Do they differ at a lot of positions? So what we do is every time we do that, we're going to get a different collection, right? We're doing the lucky draw. And every time we do the lucky draw, we're going to get a different collection of characters. Every one of those, every time we do that, we can use the characters that we get, which is the same size, same number of samples or number of characters as our original data, and we can estimate a tree. And what we can ask is, do the trees that we get by resampling in this way resemble the tree that we got from our actual data? And it may be that they do, and it may be that they don't, and it may be that they do in almost every point, except maybe this one branch point that it seems to differ a lot. Okay, so this is our way of estimating how reliable a tree is. This is the basic idea. So imagine here we have, this is a graphical description. Okay, here we have some original data on the left and one bootstrap sample on the right. So the original data has 1, 2, 3, 4, 5, 6 characters. That's the sites, the columns. If you take a look at this, a couple of the characters are identical, right? So character 1 is the same as character 4. So we have 2 of that. And the other ones are all different from each other. Some of them have mutations. When we create a bootstrap sample, what we do is we randomly draw 6 characters. So we draw a character and it becomes a character here, and we draw another character, or we draw a second character, maybe the same one, it becomes another here. So in this case, the first thing we draw is character 3, right? So that becomes character 3 is first, then character 2, then either character 1 or character 4, and then character 1, 1 or 4, character 5, and character 4, right? Character 1 or character 4. So we have here now three sites that are identical instead of 2, and we're missing one. Which one are we missing? Well, we're missing the last one, the 6th character. Okay, so we don't have the 6th character in this, but we have 3 instead of 2 of the AAAA. This is a bootstrap. So the original one, remember the bootstrap estimate or the bootstrap sequence is the same size as the original data. And it's composed of the same characters, but maybe it's going to have some characters more than once and other characters not at all. So then what happens is we get a tree, and we simply label every position on the tree with a number. It's often written as a percentage. What percentage of the time are the trees the same? And when you see numbers in the ranges of like 90%, 100%, that means that those trees don't change much. But if you see a branch where the bootstrap confidence is 25, that means when they did 1,000 bootstrap sequences, it was only 25% of the time was that branch as it is observed. And so the other 75% of the time, it's something else. So that part of the tree is less reliable. We know less about it. We have less certainty about that part of the tree. We'll see this in the practical. So that's just a brief introduction to the idea of bootstrapping. The next thing that we should discuss is the idea of rooting a tree. So some tree methods give us unrooted trees. In fact, most tree methods are going to give us unrooted trees, except for UPGMA. And as we've seen, UPGMA has a cost because we have to make this very large assumption, which is what we use as a molecular clock. So there are several ways to root a tree. Here's a couple here. So there's the outgroup approach, which we used in the forensics example and is used all the time. It's probably the most common way. We know the answer. We know that the outgroup is not in the group of interest. And so we expect that part of the tree to be separate from the part of the tree that is of interest. And we just cut the tree where the outgroup is on one side and what we're interested in is on the other side. And that becomes the root of the tree of what we're interested in. The molecular clock approach is what's used in UPGMA, and that makes a very strong assumption that's probably wrong in many cases. And that assumption is that the rate of change on all the characters is the same. And that means that there should be a point at the end where everything is equidistant to the tips. Actually, the PAM matrix construction, even though it was parsimony, it also used a molecular clock. Okay, so let me stop this video here. I will do another short video on lateral gene transfer. So I want to keep these videos short. I think it's more fun that way, and you don't open the video viewer and see, I don't know, a 60-minute video. Okay, so let me do this now and then come back and we'll do the other one.